So today we're going to finish up talking about the regulation of mean arterial pressure. So before we move forward, to jog our minds about last week, we need to take a second to break down a definition of the baroreceptor reflex. Now this doesn't have to be that whole flow chart we went through, but it can be. The basic idea of what's going on. Anybody want to give it a try? Okay. So what is the baroreceptor reflex a response to? Does anybody remember that part? Yeah, so we have these baroreceptors that are sensing differences in mean arterial pressure because of differences in the descending pressure. Specifically, when we talk about the baroreceptor reflex, what we're really talking about is this negative feedback loop in response to the drop in blood pressure. So I'd probably say something like, yeah, feedback loop. It's largely happening by upregulating the sympathetic nervous system. So increasing things like stroke volume, heart rate, and CPR with the sympathetic nervous system. We saw that there were a lot of sort of intermediate effects, ways we bring the pressure back up. But the fundamental idea is just that sharp drops in blood pressure are going to have to bring them back up because this is something that's regulated. This is homeostasis in action. That was really the whole kind of point of what we did on Friday with this big chart, right? So we were charting all the effects that are going to arise when we have this drop in blood pressure. How do we have a negative feedback loop bringing that blood pressure back up? So ultimately, because mean arterial pressure is just heart rate by stroke volume, which together are cardiac output, times total peripheral resistance, we have a series of factors that affect those three things to bring our blood pressure back up. Okay. That was our reflex. Today we're going to be talking a little bit about long-term regulation. This discussion is going to be relatively brief, but we do want to know some other ways that blood pressure is regulated. So that baroreceptor reflex that we were talking about last week, that's more of a short-term solution. It happens on a short, quick time scale compensating for a change in blood pressure, but it hasn't really corrected a problem, right? Like we've narrowed arterioles, bringing up resistance. So we've corrected the blood pressure, the mean arterial pressure for now. But whatever the problem is still a problem, right? Like we've just compensated for it. So for long-term blood pressure regulation, what we actually do is regulate the blood volume, right? So this is because like reasons for a drop in blood pressure might just be you stood up, but they might be things like hemorrhage, right? So those are the types of things we need to long-term correct. So in order to do renal regulation, we're going to use hormones, basically. And these hormones are also going to have effects on some of the parts of the cardiovascular system that we've been talking about in this. So the three we're going to pay attention to are epinephrine, vasopressin, and angiotensin 2. So epinephrine in specific should not be a huge surprise because we've already talked about the fact that epinephrine is acting on the arteriole, right? We talked about epinephrine and how it's affecting alpha receptors, but also beta 2 receptors, right? We talked about how it acts on the smooth muscle of the arteriole. So our overall effect of the epinephrine is going to be to increase total peripheral resistance. Also saw that epinephrine is acting on the smooth muscle of the vein. So increasing venomotor tone, essentially increasing how tense those venous walls are, and therefore raising pressure in those veins going into the heart, therefore increasing how much blood gets into the heart, how fast it fills. And then epinephrine acts on the heart itself as well. So epinephrine released by the adrenal glands is paired with activity of the sympathetic nervous system usually, right? Because the sympathetic nerves are going to the adrenal gland in order to make it release epinephrine. So epinephrine, when it's circulating in your bloodstream, is going to have similar effects on the heart that sympathetic innervation of the heart itself did. So it's going to increase heart rate, and it's going to increase stroke volume because it's affecting those nodes and the contractility of the heart in the same way that the norepinephrine coming out of the sympathetic nerves would. For vasopressin and angiotensin 2, what we want to know about for the purposes of understanding regulation of mean arterial pressure is that they're vasoconstrictors. They're going to cause our blood vessels, our arterioles to constrict. We know that our equation for understanding properties that affect mean arterial pressure, as we had our mean arterial pressure equivalent to stroke volume by heart rate, which is cardiac output, times total peripheral resistance. Okay. Because vasopressin and angiotensin 2 raise total peripheral resistance through vasoconstriction, right? Because as the radii of these vessels are decreasing, their resistance is increasing. This means we are also raising our mean arterial pressure. So at the same time as those things are happening, we would have long-term regulation happening with the kidneys, and we'll go into detail about that in our renal exam, which is not the next one, but our third. So the baroreceptors we've talked about so far were the ones in the aorta and the carotid sinus. They were arterial baroreceptors, so they were sensing stretch and descending pressure in the arteries. We also have baroreceptors in the heart itself, in the atria, and in large systemic veins. So these are our cardiac and venous baroreceptors. So they also sense pressure. They also do this by essentially sensing stretch. But we often refer to these ones as low pressure baroreceptors or volume receptors because of their location. So in the atria and in the systemic veins, we don't really have a ton of pressure, right? So that's where the low pressure baroreceptors come from. So what we're really sensing there kind of is how much blood there is. So how much that atrium is filling, right? And just what the state of the walls of the systemic veins is, like how expanded versus not expanded they are. Otherwise, they're going to work in concert with our arterial receptors. So when we have a decrease in blood volume, it's really going to correspond to be happening at the same time as we have a drop in mean arterial pressure, that's by the arterial baroreceptors. So they're going to be working kind of in parallel to trigger these responses that go along with the baroreceptor reflex. So this is just kind of another place that we can sense these things happening. A separate set of receptors doing similar things. Okay. Next, we have our kind of grab bags of effects on mean arterial pressure. So we're going to talk a little bit about how breathing, chemical content of your blood, temperature affect some of these factors that we've been tracing. Okay. So the concept of respiratory sinus arrhythmia. Arrhythmia is like a difference or change in rhythm. What this is saying is we're observing the fact that some people's heart rate and their nervous activity actually changes depending on whether they're breathing in or out. So like this is why people taking a shot right will often do it while they breathe out, because this slows down your heart rate due to activation of the parasympathetic nervous system during expiration, which means you're a little steadier because your heart rate's not racing. And the converse happens during inspiration. So when you breathe in, there's actually a little bit more sympathetic activity. Those sympathetic nerves from the autonomic nervous system are active. And the sympathetic nerves are the ones that do the fight or flight, speed up heart rate. So this has an effect on your heart rate, and therefore slight effects on things like mean arterial pressure at this point. So breathing in, your heart rate speeds up a little. Breathing out, your heart rate slows down a little. So the arrhythmia is because we don't have necessarily this steady, exactly same heart rate during breathing in versus breathing out. Next, we have our chemoreceptor reflexes. So we're going to talk about chemoreceptors a fair bit as we talk about the respiratory system. But what we want to know now is that we have chemoreceptors so that sense concentration levels, specifically mainly of carbon dioxide. We have sensors that tell us, do we have too much carbon dioxide in our blood? So what these are doing in general is trying to regulate those blood carbon dioxide levels, bring them back down, because carbon dioxide, as we'll see in our respiratory unit, actually has a big effect on blood pH as well, which will become important to us.
Are chemoreceptors in the lungs, or are they all throughout the body?
Chemoreceptors are in the brain. So these chemoreceptors are responding to an increase in carbon dioxide. So that's that first bit here. And their effect overall is to increase total peripheral resistance and decrease heart rate. Now, initially, if you kind of think about the problem they're trying to solve, this isn't so intuitive, right? If we think too much carbon dioxide, we want to get rid of the carbon dioxide, right? If we decrease the heart rate, then we'd be decreasing flow. So what's actually happening here with the chemoreceptors is that we're decreasing the heart rate, not so much with the goal of changing the flow, but to change the metabolic needs of the body itself. So by decreasing the heart rate, slowing it down, that heart doesn't need so much energy, doesn't need so much ATP, so metabolism actually slows down a little bit. So the way that we are decreasing the carbon dioxide in the blood level by decreasing heart rate is by producing less CO2. At the same time as that, we have total peripheral resistance increasing. So overall, we have a result of a slightly increased blood pressure, increased mean arterial pressure, I should say. And this is also helpful because if we remember, we also saw that the amount of carbon dioxide has an effect on the smooth muscle around the arterioles themselves, right? We saw that they tend to dilate in response to buildup of CO2. So if we have more CO2 out in the body, those arterioles may be beginning to dilate. And then this chemoreceptor X can clamp that down, basically increase the resistance of it so they don't dilate too much and don't drop our blood pressure too far down. Okay. Next is our thermoregulatory response. So this has to do with temperature regulation. An interesting thing is that temperature regulation actually takes precedence over blood pressure regulation. So both of these are going to be homeostatically controlled mechanisms, right? They're both things your body is trying to regulate. It cares more about your temperature than it does about your blood pressure. So thermoregulation, coming from the hypothalamus, that's kind of our integration center for body temperature. What happens when we have an increase in body temperature, we have a decrease in the sympathetic activity to the skin. The sympathetic activity had a tendency to constrict things, right? Had a tendency for vasoconstriction through sympathetic activity. So when we decrease sympathetic activity to the skin, we have the opposite effect. So we have vasodilation. So our temperature went up, we decreased sympathetic activity. This allows our blood vessels to dilate, to get bigger, to have more blood flow to the skin. So this is helpful for your temperature regulation, for your thermoregulation, because this means you have your blood in the capillaries, basically at the border with the environment, coming into contact with the environment. I mean, you're not bleeding, but they're in the skin there close. So we're able to have an increase in heat loss from your blood out into the environment. So heat is dissipating from the skin into the air around you. And at the same time, we like to have more sweating, also assisting in an increase there of heat loss to the environment. Now the problem with this, right? So our mean arterial pressure is equivalent to our heart rate, our volume, and our total peripheral resistance. Okay, so if we have vasodilation happen, is total peripheral resistance going up or down? Yeah, exactly. Yeah, it says it right there on the slide, not trying to trick you. So dilation was we made the radii of those blood vessels larger, which means we had decreased resistance. So now we have dropped down our total peripheral resistance. We can see how this would have a tendency to drop down blood pressure. And this might even be an increasing effect if we're sweating a lot at the same time, because then we're also losing fluid. And we know that the amount of fluid in the cardiovascular system is part of what keeps our blood pressure here, right? So we may also be decreasing the amount of blood, which could kind of have an effect on stroke volume. Maybe that's where I draw it in. Not a direct effect on these factors, but overall, we may have a decrease in mean arterial pressure, which is part of why you can get heat sickness, right? Like part of why you feel those effects is this change in blood pressure in response to trying to thermoregulate. We just have a reminder about how all these various factors go together when we think about exercise, because exercise is something that really changes the demands on our cardiovascular system. So if we're focusing in, we might focus in here. So we've been talking about mean arterial pressure. You can see that there's a slight rise in mean arterial pressure during light exercise. But we can see we've had a huge increase in cardiac output, which is one of our factors adding into arterial pressure. So we have had also a decline at the same time in total peripheral resistance, kind of offset this. All right, so the reason we have an increase in mean arterial pressure is that our overall effects on heart rate and stroke volume, that's our cardiac output, are bigger than the opposing effect on total peripheral resistance. So these things are balancing each other for the most part, with the result of just a smaller increase in mean arterial pressure. We can see heart rate has changed a lot. Stroke volume has changed a bit. And we know that at the same time, a lot of our effects on resistance are happening kind of at an organ by organ level. So we have a lot of cardiac output, but most of that is being routed to the muscles and to the heart muscle itself. Whereas in the rest of our system, so for example, in our gut, we are decreasing blood flow to those organs, which is why we don't have an even bigger effect on the total peripheral resistance. Okay, do a couple of questions, and then we're going to start talking about the blood itself. Okay. For this question, we are looking at mean arterial pressure falling and the baroreceptor reflex getting activated. That's at negative feedback loop. So then we have some factors that may or may not be associated with the reflex. Okay. So our options are decreased compliance of the veins, vasodilation of the arterioles, decreased stroke volume, and increased parasympathetic activity. I'm going to start from the bottom up for once. So remembering that our baroreceptor reflex, right, this is our negative feedback loop. So what we're trying to do, right, our mean arterial pressure has dropped, and we're trying to bring it back up. So we know that our effects are going to act on the heart, either on the heart rate, the stroke volume, or both. And we're also going to act on the total peripheral resistance. And this is just another different way to look at it. You can also just look at the flow chart. So starting from the bottom up, for increased parasympathetic activity, who remembers what happens when our parasympathetic nervous system is active? Yeah, you relax, that's the rest and digest. So that would have a tendency to do what to heart rate? Yep, lowers heart rate. So this is going to lower our mean arterial pressure. Decrease stroke volume. You can already see what it's going to do from this equation. Right, so that would also lower mean arterial pressure because of the effect on stroke volume. Which of these factors would vasodilation of the arterioles affect, do you think? Yep, total peripheral resistance. And what would it do? Yep, as we dilate arterioles, we would be decreasing our total peripheral resistance. So by process of elimination, we've gotten to decreased compliance of the veins. We might also have gotten there from remembering our flowchart, remembering these effects. But overall, to remember this idea of compliance is that like this trade-off between pressure and volume. And we can just remember that our veins generally have high compliance. Arteries have low compliance. Right? So what we have done in order to increase pressure in the veins and therefore increase blood flow back to the heart is to make those veins a little bit more like arteries. We've made them a little more tense. That's that venomotor tone. And so we have decreased their compliance. if any of you guys have a real good gut sense of compliance, I highly recommend or would ask you to explain it to each other as many times as many ways as you can, because I think it's a tricky thing to kind of just like get a gut level understanding of. So I'm trying to say it in different ways, just hoping any one of them will click. But sometimes when you're working with people who are learning stuff at the same time as you, it can come up with tips and tricks that I don't know, that I can't think of right now. Okay, so it's decreased compliance. Okay, so here's a question from today about the thermoreceptors and the hypothalamus. Okay, so we go through this the same way. So we've said body temperature rose. All right, so heat rises. So in response to heat rising, we're going to want to bring body temperature back down. Sweating actually reduces temperature, so this is not the case. This would make you hotter, right? So not that one, right? We'd increase sweating in order to bring temperature down. Decrease vascular resistance to the skin. What's another way of saying decreasing vascular resistance? Can anybody phrase that differently? No, not the veins, but the blood, yeah. Yeah. And so that's why, what have we done to the radius of the arterioles of those blood vessels? Yeah, exactly. So this is really just describing vasodilation. That's a description of vasodilation, which is bringing more blood to the surface, allowing us to thermoregulate. So now we did just talk about that. So it would actually have the opposite effect of vasoconstriction. It's not really to do with the cardiac output here. That's how we get to answer B. We're going to get started on the blood, and we will be skipping some of the content that's in this packet because hopefully you have looked a little bit, especially at white blood cells in lab last semester, I believe, in physiology. So what we're going to focus on for our purposes is understanding the life cycle of erythrocytes, of red blood cells, how hemoglobin works. So we're going to be thinking about how oxygen and carbon dioxide circulate around. And then we're going to think about clotting. So those are our three major topics that we're going to really be focusing on here. I don't think I'm going to test you on them for this exam, frankly. Yeah. But since if you have printed out your slides, they're already going to be there. I'm just like letting you know. So I'll probably just tell you to skip slides as we go through. Okay. So we're trying to understand the components of the blood. So we will be focusing in, for our purposes, in this big packet so that we don't have too much because we only have one day left before our review. We're going to be focusing in on our erythrocytes, and then we'll be focusing in on platelets and hemostasis, which is really going to be clotting. And that has effects on these different conditions. I'll run through these really quickly. So hopefully these are terms that are familiar to you from physiology lab. Hopefully remember that hematocrit refers to the percentage of blood volume that's made up of our erythrocytes, those red blood cells. Remember that our plasma mainly made up of water and a lot of different plasma proteins. one of the really important ones in terms of creating osmotic pressure, so therefore influencing things like net filtration pressure, which we focused on with the capillaries, is one called albumin. Hopefully you spoke about that in physiology lab last term. So you've probably seen this slide before. So we have average blood volume of 5 to 5.5 liters. 3 liters of that is plasma, so 3 liters is fluid. Then we have a hematocrit that differs a little bit between men and women, but is a little bit under 50%. So that's our erythrocytes, our red blood cells. And then we also have a buffy coat, so a small layer of leukocytes, our white blood cells, and platelets, which are kind of like cell fragments that we'll talk about a little bit as relates to blood clotting. Here we can see just a sort of general picture of how blood cells develop. So we start with hematopoietic stem cells. So these are going to be like in bone marrow, basically, and they divide into different types of stem cells, next myeloid or lymphoid, and ultimately, we're not memorizing the inside levels, but give us our types of blood cells we're interested in. So what we are going to be focusing on is our red blood cells. So we might trace this line, right? So basically what we would want to know about the red blood cell, about its development that is kind of interesting, kind of unique, that we start with these hematopoietic cells. We develop all our components of the red blood cell, but then you can see that we actually lose most cellular components before we create adult mature erythrocytes. Okay. We can see that these erythrocytes, these red blood cells, are flat, they're lacking things like mitochondria, they're really small. So they look fairly different than a lot of our other types of blood cells. Okay. And we'll also see that as we think about different conditions, we may measure whether we can see any of this sort of progenitor cell in the bloodstream, these reticulocytes that still have some of those components. Because if you're producing red blood cells really quickly, you may see some of these in the bloodstream as well. Okay. Our plasma, almost entirely water, like 90%. We have some protein, 6 to 8%. And we have relatively high concentrations of some of our ions, specifically sodium, which we've looked at a lot in previous units, but also a little chloride ion. And then we have some other sort of trace ions, electrolytes. So we have hydrogen ions, carbonate ions, potassium, calcium. We've seen a lot of these before as we've talked about different body processes, right? We talked about sodium and potassium and calcium as we talked about neurons. We talked about some of these in relationship to the heart rate as well. We talked about the cardiac cycle and what's going on the cellular level there. We looked at hydrogen ions way back when we talked about metabolism, right? That part of the way we're producing ATP is with a hydrogen ion gradient. We'll see that.
So these are all free floating ions in the plasma?
These are in the plasma. Yeah. Well, both. Yeah. For now, we're talking about free floating, but yeah. So our erythrocytes are red blood cells. You have about 5 billion of them in a single milliliter of blood. They're shaped kind of like a frisbee. They're a biconcave disc, which is to say, I don't know, like maybe like a Cheerio with the inside filled in, right? So they're, oh, now that I'm doing this, I think it looks like a sweet tart, whatever. Concave on both sides, kind of flat in the middle, these bulging edges of the side. And this is helpful for us because this really expands the surface area of a red blood cell. So there's not much of a diffusional barrier here. So this is going to facilitate oxygen getting into those red blood cells and getting onto the hemoglobin in the red blood cells. So this is part of why it's important that they're small and thin and they're this specific shape. So their diameter is about 8 microns, thickness about 2. And I think you did some math problems about stacking them up in this. We will not be doing math about red blood cells in lecture, which I am happy about. But they are very small. Their membranes are flexible, and a specific characteristic trait of our red blood cells are erythrocytes, that they no longer have a nucleus and they no longer have organelles. So we needed a nucleus and organelles while we were making the red blood cells in order to get largely the hemoglobin, but other components. But these organelles in the nucleus have atrophied, deteriorated, have died away in order to make our red blood cells really nice and small and compact, pretty much just full of chemicals that we're going to need for gas exchange. So they have no mitochondria. They have no anaerobic glycolysis. I think that means only anaerobic glycolysis. Regardless, they can't make very much ATP. Their major function is really just to transport oxygen and carbon dioxide, and we're going to be regularly making new red blood cells. So we're going to be regularly replacing them because they don't have many organelles and can't do all sorts of different repair functions. They're going to be kind of short term. The most important protein inside of our erythrocytes, our red blood cells, is hemoglobin. So hemoglobin is really 4 polypeptides together. We have subunits, which we could trace. So here we have an alpha subunit. Here we have another alpha subunit. And then at the back, we have 2 beta subunits. So we have these 4 subunits that have come together to make one protein. And within each of these subunits, we're holding a heme group. So the globin part is like a protein polypeptide part, and these heme groups contain iron. So this is where we're going to be doing much of our oxygen transport. So almost 99% of the oxygen that we transport in the blood is bound to hemoglobin. So most of it is stuck onto a heme group. There is some dissolved in the plasma itself, which is important because we are going to have to release oxygen from the hemoglobin in order to get it into a cell. We're not going to be transporting hemoglobin molecules in. We're going to have to let go of the oxygen and let it dissolve across our capillary walls into tissue. So there is a little bit of O2 that's just dissolved directly in the plasma, but most of it at any given point is stuck onto one of these heme groups. When hemoglobin is oxygenated, so when we have an oxygen molecule bound into this heme group, and specifically bound into this iron, right? There's iron in the center of the heme group. Blood is bright red. So this is why we talk about arterial blood as being bright red. And essentially, you know, if you're taking blood, you hopefully won't hit bright red blood. You shouldn't. So when you hit this bright red blood, it tells you it's oxygenated, tells you you're in an artery. Deoxygenated blood is dark red. So not necessarily actually blue, right? But that's our veins, our venous system, actually changes color, like physically changes color as oxygen jumps off of our heme groups. When we think about hemoglobin, we're mostly thinking about oxygen transport, but that's not actually the only thing it's doing. So it's also binding carbon dioxide, some proteins, and carbon monoxide, which is a little unfortunate. It actually binds carbon monoxide better than it binds oxygen. So it is doing these other things as well, not just oxygen, but oxygen is its like main role. For our purposes, specifically, we don't want to forget about this carbon dioxide effect though, because we are separately going to be tracing what's happening to carbon dioxide in the blood. So it's important to remember some of it's going to be dissolved in the plasma, some of it's going to be on another protein that we're about to talk about, some of it's going to be on the globin parts of the hemoglobin actually. So carbon dioxide and oxygen aren't fighting for these heme groups. The oxygen is on the heme group, and then the carbon dioxide would be like somewhere else on the polypeptide part. So the other thing our red blood cells, our erythrocytes, are going to have is carbonic anhydrase. Carbonic anhydrase is another important protein and enzyme that is going to be in our blood that's going to be critical to the transport of gases. So what this enzyme does is it allows us to take carbon dioxide and water and turn it into carbonic acid. That's carbonic acid. And carbonic acid then can separate into hydrogen ions and bicarbonate ions, which is going to basically create pH balance in the blood. So the amount of hydrogen versus bicarbonate in the bloodstream is going to create the pH of the blood. So we'll talk about in the future how that's regulated through respiration and also through the renal system. Okay. So the carbonic anhydrase is important to transporting carbon dioxide, partially because we can take that carbonic anhydrase and turn that carbon dioxide into carbonic acid. When we talk about carbon dioxide in the respiratory system, we're sometimes going to refer to it as like a biological acid because it has this property of being able to be turned into carbonic acid, which then dissociates. So the levels of carbon dioxide are going to be really critical to maintaining acid-base balance in the blood. Now take a second to make sure we remember the big difference between red blood cells and other cells. Big difference is that we lack these typical cell structures. No nucleus, no mitochondria, organelles, which means we can't make proteins, which means there's a lot of stuff a red blood cell cannot do. Remember the function of hemoglobin. And we're going to remember that combines both oxygen and carbon dioxide. So different parts of the hemoglobin, but both involving this molecule. Okay. So now we're going to talk about the life cycle of erythrocytes as relates to the cardiovascular system, but also organs. So we're going to be like physically kind of tracing them from where they're produced to where they're basically eaten up for their component parts. So the life cycle of an erythrocyte is different than a lot of other cells because they can't undergo mitosis. Like most cells in your body, right? You have one cell, undergo mitosis, cell division, and split into two identical cells. Right? That's typical. Your skin cells can do that. But this requires the ability to take, well, first off, to have DNA in a nucleus, to replicate that DNA, then to create all sorts of extra organelles so that you can parcel them out into two cells. And the red blood cells don't have any of these components. They just can't do it. Right? They don't have the nucleus, so they don't have the DNA. They can't make any type of protein because they don't have any DNA to do it. And even if they did, They don't have ribosomes in order to make those proteins. So erythrocytes cannot make more erythrocytes this way. You can't take a red blood cell and make it make 2 red blood cells. It won't do it. But we do still need to replace our red blood cells. They have a lifespan of about 120 days, and you actually replace about 2 to 3 million red blood cells per second. Okay? Because they get old and worn out just like any other cell. So what happens instead of mitosis to replace these red blood cells that you're getting out of circulation, is we're going to be synthesizing them newly in red bone marrow through erythropoiesis, kind of all the time. And then we're going to be filtering them out using the spleen and a little bit using the liver. We'll take a look at that in a bit. So they're going to have this life cycle that is going to involve our bones, our bloodstream, and the spleen and the liver. And we're going to see that part of the reason for this life cycle, especially this part with the spleen and the liver, is we're going to want to hold on to things like that iron in the hemoglobin for reuse, right? So just because we're replacing the red blood cell doesn't mean we want to get rid of absolutely everything that was in that red blood cell. Okay, so we're going to stop there and we'll pick up with the synthesis of erythrocytes on whatever day it is, Wednesday.